Hello, my name is Churishma Chandu, and this video is going to be about action molecules and two models, the lock and key model and the induced fit model. First, let's look at the different parts of these two models. This green shape here is the substrate, this blue circle is the enzyme, and this white space in the enzyme is the active site for the lock and key model. In the induced fit model, the blue circle is the enzyme, the white space is the active site, the red circle is the substrate, and the pink cone is the coenzyme. So how do these models work? The substrate starts to enter the active site of the enzyme in stage 1. In the lock and key model, the shape of the substrate is complementary to the active site. But in the induced fit model, as you can see here, the shape is not the same for the substrate, coenzyme, and the active site of the enzyme. In stage 2, the enzyme grabs the substrate into the active site to bind. This is called the enzyme-substrate complex. In stage 3, the catalysis happens. Catalysis is a chemical agent that selectively increases the rate of a reaction without being consumed by the reaction itself. This is called the enzyme products complex, where the substrate completely binds to the enzyme. In the lock and key model, the substrate binds to the active site without any change since they are complementary. But in the induced fit model, the substrate and the coenzyme binding to the enzyme's active site results in a conformational change to the enzyme. In the last stage, when the enzyme releases the products, the shape of the active site of the enzyme goes back to its original shape so it can be ready to work on another chemical reaction. As you can see here, the red circle and the pink cone and the red shape and the pink triangle here are the products of the models. So how can enzymes be so specific in terms of the substrate they impact? In the lock and key model, enzymes are substrate specific, which means that the enzyme can only work with one substrate or a set of reactants. This is because the active site, the site on the enzyme where the substrate binds, and the substrate itself are complementary in shape. In other words, they fit together like how a key fits in the lock. In the induced fit model, when there is a substrate present, the active site undergoes a change in its conformation to allow a better fit between the active site and the substrate and coenzyme sometimes. This model doesn't have a complementary active site for a specific substrate to fit. So the active site shape of the enzyme can be modified for the binding of the substrate. The induced fit is caused by the entry of the substrate, the change in the shape of the active site of an enzyme so that it binds more snugly to the substrate. What are coenzymes? Coenzymes are organic molecules that bind to the active site of an enzyme and participate in catalysis, but are not considered substrates. Sometimes coenzymes help the substrate and the enzyme's active site bind. There's a coenzyme in the induced fit model on the right here. It is the pink cone under the red circular substrate here. It helps the substrate bind to an enzyme. It also functions as a catalyst to speed up the chemical reaction.